This video tutorial is about the fixation of carbon dioxide and the light-independent stage of photosynthesis. The light-independent stage of photosynthesis takes place in the stroma of chloroplasts. Although it does not directly use light energy, it uses the products of the light-dependent stage. If the plant is not illuminated, the light-independent stage soon ceases, because ATP and hydrogen are not available to reduce the carbon dioxide and synthesize large complex organic molecules. Carbon dioxide is the source of carbon for the production of all organic molecules found in all the carbon-based life forms on Earth. These organic molecules may be used as structures, such as cell membranes, antigens, enzymes, muscle proteins, or cellulose cell walls, or act as energy stores, such as starch and glycogen. Carbon dioxide in air enters the leaf through the stomata and diffuses through the spongy mesophyll layer to the palisade layer, into the palisade cells through their thin cellulose cell walls, and then through the chloroplast envelope into the stroma. The fixation of carbon dioxide in the stroma maintains a concentration gradient that aids this diffusion. Carbon dioxide, that is a byproduct of respiration in plant cells, may also be used for this stage of photosynthesis. The series of reactions whereby carbon dioxide is converted to organic molecules is called the carbon cycle. I'm going to go through the steps of the carbon cycle now. Step number 1. Carbon dioxide combines with a carbon dioxide acceptor, which is a 5-carbon compound called ribulose biphosphate, represented there as RUBP. This reaction is catalyzed by the enzyme rubisco, or ribulose biphosphate carboxylase oxygenase. Step 2 is RUBP. By accepting the carboxyl group, which is the COO- group, it becomes carboxylated, forming an unstable intermediate 6-carbon compound that immediately breaks down. Step 3. The product of this reaction is 2 molecules of a 3-carbon compound represented as GP, glycerate 3-phosphate. The carbon dioxide has now been fixed. Stage 4. GP is then reduced using hydrogens from the reduced NADP made from the light-dependent stage to triose phosphate, or TP. Energy from ATP, also made from the light-dependent stage, is used at this stage at the rate of 2 molecules of ATP for every molecule of carbon dioxide fixed during stage 3. Step 5. In 10 of every 12 TP molecules, the atoms are rearranged to regenerate 6 molecules of RUBP. This process requires phosphate groups. Chloroplasts contain only low levels of RUBP, as it is continuously being converted to GP, but is also continually being regenerated. The remaining 2 of the 12 molecules of TP are the product. As already mentioned, the products of the light-dependent stage, namely ATP and reduced NADP, are continuously needed for the Calvin cycle to run. During the light-dependent stage, which is shown on the slide in front of you, hydrogen ions are pumped from the stroma into the thylakoid spaces So the concentration of free protons in the stroma falls, raising the pH to around 8, which is optimum for the enzyme Rubisco, which is represented as RuBisco. Rubisco is also activated by the presence of extra ATP in the stroma. In daylight, the concentration of magnesium ions increases in the stroma. These ions attach to the active site of Rubisco, acting as cofactors. The ferredoxin that is reduced by electrons from the photosystem 1 activates enzymes involved in the reactions of the Calvin cycle, which is shown again on the slide in front of you. I'm just going to finish by mentioning a couple of the uses of triose phosphate, that's TP. Some TP molecules are used to synthesize organic compounds. For example, some glucose is converted to sucrose, some to starch, and some to cellulose. Some TP is used to synthesize amino acids, fatty acids, and glycerol. The rest of the TP is recycled to regenerate the supply of RuBP. 5 molecules of the 3-carbon compound TP interact to form 3 molecules of the 5-carbon compound RuBP.